Hello my dear students, welcome back to Biology at Priyajit. Today's topic is Leishmania donovani, one of the most important protozoan parasites in medical parasitology, microbiology and zoology. This parasite basically causes Kala-azar, also known as Visceral Leishmaniasis, one of the most severe parasitic diseases affecting humans. In this lecture we shall discuss introduction, classification, disease and vector, morphology, life cycle, pathogenesis, clinical symptoms, diagnosis, control and treatment. So let's begin. Leishmania donovani, the disease is Kala-azar or visceral leishmaniasis. The vector is female sandfly or Phlebotomus species. Morphological forms, Leishmania exhibits two morphological stages. First one is the Amastigote form. The characteristics is this is oval, round to oval body. Basically they are non-flagellated, intracellular. They are found inside the macrophages. They have flagella sometimes, but this is short and embedded. There are multiplication stages in humans. So this is the Leishmania mastigotes phagocytosed by macrophages, and the black arrow shows the amastigotes. The amastigote is basically the form found inside the human body. It lacks a free flagellum and therefore they cannot swim. The flagella is embedded and short. Instead, it survives inside macrophages. This is the image showing their presence within macrophages, escaping destruction and multiplying by binary fission. This image showing the fundamental life cycle of the Leishmania donovani. The second stage is the Promastigote form, which is elongated, spindle-shaped, flagellated, motile. They found in sandfly and they are infective stage. So the Promastigote possesses a long anterior flagellum that helps in movement. This is the infective stage for humans because it is injected by the female sandfly during blood feeding. This is the Promastigote stage of Leishmania donovani 100x and are stained with GMs are stained. So the life cycle of Leishmania, it has a digenetic life cycle. Life cycle in human, the infected female sandfly bites human, injects the Promastigote into human body. Promastigotes enter the bloodstream of the human. Then within the bloodstream, the macrophages engulf Promastigotes. Inside the macrophages, Promastigotes transform into amastigote. And then the amastigotes multiply by binary fission. Large numbers accumulate, then they rupture the macrophages, release amastigotes, infect new macrophages, and the cycle repeats. This is the life cycle of the Leishmania donovani. Notice that multiplication occurs only in amastigote stage, and this continuous destruction of macrophages leads to enlargement of organs rich in macrophages, particularly spleen, liver, bone marrow. Life cycle in sandfly: when another female sandfly feeds on an infected person, it injects macrophages containing amastigotes. So inside the sandfly, amastigotes transform into promastigotes, then multiply rapidly, migrate to the proboscis and ready to infect another human. So now what is the pathogenicity? The parasite primarily attacks liver, spleen and bone marrow. These organs contain numerous macrophages. As infected macrophages rupture repeatedly, normal immune function declines, blood cell production becomes impaired and the organs enlarge significantly. Without treatment, visceral leishmaniasis can be fatal. This is the image showing the pathogenesis of Leishmania. Clinical symptoms. The most striking feature of Leishmania is massive enlargement of the spleen. Because bone marrow is affected, patients develop severe anemia and weakness. The progressive darkening of skin gives rise to the name Kala-azar, meaning black fever. So the clinical symptoms are irregular fever, massive splenomegaly, enlarged liver, anemia, weight loss, weakness, darkening of skin. Untreated disease is frequently fatal. So this is the image showing the mnemonic for proper remembrance of the students about the symptoms caused by Leishmania donovani, or which gives rise to the Kala-azar disease or black fever. Diagnosis relies on identifying the parasite directly or detecting the parasite-specific antibodies. Their molecular methods such as PCR provide high sensitivity, especially in difficult cases. Laboratory diagnosis, demonstration of amastigotes in spleen, bone marrow or lymph node aspirates. Serological tests like RK39, rapid diagnostic test, PCR-based molecular diagnosis, culture in specialized media, which is less commonly used. Control measures include sandfly control, insecticide spraying, elimination of breeding places, protective clothing, mosquito or sandfly nets, early diagnosis and prompt treatment. It is recommended drugs include liposomal amphotericin B, miltefosine, paromomycin, etc. Among these, liposomal amphotericin B is considered one of the most effective treatments for visceral leishmaniasis due to its high efficacy and comparatively lower toxicity. So this is the summary. Today we learned Leishmania donovani causes Kala-azar or visceral leishmaniasis. It has two morphological forms, Amastigote and Promastigote. The parasite alternates between human and female sandflies or Phlebotomus. It multiplies inside macrophages, leading to damage of the liver, spleen and bone marrow. Common symptoms include irregular fevers, splenomegaly, hepatomegaly, anemia, weight loss and skin darkening, which is called as black fever or Kala-azar. Early diagnosis, vector control and treatment with liposomal amphotericin B, miltefosine or paromomycin are essential for reducing disease burden. So my dear students, if you found this lecture helpful, please like, share and subscribe to Biology at Priyajit. Don't forget to press the bell icon so you never miss our detailed lectures on parasitology or medical zoology or other topics of zoology, microbiology for UG and PG students. Thank you for watching. I'll see you in the next class. Bye.